Hello my dear students, welcome back to the channel BioPulse, and this is Chandrika. Today we are going to discuss a very beautiful and then very interesting chapter, that is molecular basis of inheritance and part 1. Because this chapter is big and then very interesting chapter, so I am making two parts of this, and today is the day 17 one. Have you ever wondered how a tiny molecule inside your cell decides your eye color, height, and even certain diseases? Yes, of course, you can know all the reasons for all these in the chapter, that is molecular basis of the inheritance. The chapter where life's secret code is finally revealed. In this chapter, we will explore DNA, that is the molecule that carries genetic information, understand how it replicates, how it is forming the RNA, and how proteins are made through the process like transcription and translation. This is the core of genetics and one of the most important scoring chapter for the NEET. On an average, if I say, you can expect 2 to 4 questions from this chapter for the NEET, that is contributing up to 8 to 16 marks, isn't it? Making it a high weightage and high return topic if you prepare very smartly. So if you want easy marks with clear concept, this chapter is your golden opportunity. If I say DNA decides everything about you and NEET decides everything about your future. So let's break it down into simple tricks, clear concepts, and exam focused points so you can master it without confusion. So let's begin. This is the number 1 question for today's chapter, that is a beautiful chapter, molecular basis of the inheritance. Number 1, monomer of nucleic acids are option A, peptides, B, nucleosides, C, ribonucleosides, and then D, none of this. Here the right option is option D, none of this, because the actual monomer, that is monomer in the sense building blocks, okay, nucleic acids monomer is actually what? They are the nucleotides. Here nucleotides are not there, so the monomers of nucleic acid is nucleotides. That's what the option none of this is correct here. Second question, DNA and RNA are types of A, nucleotides, B, nucleosides, C, nucleic acid, and then D, nucleamides. Here the right option is option C, that is nucleic acid. We have two types of nucleic acid, that is DNA and then RNA. Number 3, questions from the concept, that is the DNA structure, everything you will learn much more things about here. Third question, the length of the DNA is usually defined as, A, number of nucleotides present in it, yes. B, number of pairs of nucleotide present in it, yes. C, number of base pairs present in it, and then D, all of this. So here the right option is D, all of this. See, number of nucleotides is used to get a single stranded length, isn't it? And the number of pair of nucleotides, it is used to get a double stranded DNA's length. It is mostly used. And number of base pairs, it is used for the numerical count of these units for the physical length of the DNA, or else you can go for it. So here the fourth question, match the length of DNA with the correct organism. First one here, delta 174 is given, this is the A, column A, and then column B. Delta into 174, this is a bacteriophage actually. It has nucleotide number, DNA length if I say 5386 nucleotide. Where is 5386? Yes, so 1 is 4. 1 is 4 means we have two options, either A, either C. So let me go for next one, second one, that is bacteriophage lambda. Bacteriophage lambda has 48502. So 2 is 3. 2 is 3. Again A and C has both, isn't it? So we'll go for third one, E. coli. E. coli is having 4.6 into 10 to the power of 6 base pair. BP in the sense base pair. This must be, this is not 106, it's a 10 to the power 6. Okay, so 1 is 1, 3 is 1. So right option here it is option A. Next question, that is fifth one, structure of polynucleotide chains. From this concept, fifth one, a nucleotide contains A. hexose sugar nitrogenous base phosphate group, B. pentose sugar nitrogenous base phosphate group, C. hexose sugar nitrogenous base phosphate group, and then D. pentose sugar nitrogenous base and then sulfate group. Here the right option is B. pentose sugar nitrogenous base and then phosphate group. If I draw a simple structure, there will be a pentose sugar. Just consider it as a pentose sugar. In nucleotide, a single nucleotide has pentose sugar, this is pentose sugar, and nitrogenous base. Here there will be nitrogenous base. I will simply call it as NB. Okay, and there will be a group which is called as what? Phosphate group. If the sugar, that is pentose sugar, is attached with the nitrogenous base and then phosphate group, then it is called as what? Nucleotide. Okay, so here the right option is option B. Sixth one, which of this is a purine? Which of this is a purine? A. cytosine, B. adenine, C. thymine, and then D. more than one is correct. Here the right option is option B, that is adenine. So I will tell you a trick to remember purines versus pyrimidines. Pure as gold. Purine, just remember pure as gold. As gold. A stands for adenine and G stands for guanine, isn't it? So purines include A and G, pyrimidines include CTU. Cytosine, thymine, and then uracil. Got it? How you'll remember purines? Pure as gold. Pure as gold, in that as A is indicating what? Adenine, whereas G indicate gold, G indicating guanine. Seventh one, which of these is a pyrimidine? Just now I said pyrimidine contains C, T and U. Cytosine, thymine and uracil. Among them thymine is there, so right option here it is B. Which of these is a correct combination for a DNA nucleotide? Which of this is a correct combination for the DNA nucleotide? Options: A. oxyribose phosphate plus uracil. He is asking for DNA. Uracil will be there in the RNA. Oxyribose is also for RNA. Deoxyribose phosphate and uracil, again wrong. Deoxyribose phosphate and then thymine, this one is correct. So option D is correct. DNA, in the DNA expansion, if you ask deoxyribose nucleic acid, isn't it? So in the DNA what happens, the one oxygen is reduced compared to the oxyribose, compared to the RNA, if you see the structure. So here the sugar molecule of the DNA has deoxyribose sugar, not the ribose one. Okay, oxyribose one. Got it? And phosphate group is having phosphate group and bases, and this is the pyrimidine, isn't it? So here the right option is D. Next, ninth one. All the given nucleotides exist except deoxyuridine, thymine, C both A and B, and then D none of this. Here the right option is D, none of this, because these all are the nucleotides. 10th one. Nitrogenous base is linked to which carbon of pentose sugar? Which carbon of pentose sugar? Carbon 1, carbon 2, carbon 3, and then carbon 5. Here the right option is option A, that is first carbon. When the nitrogenous base is linked to which carbon of the pentose sugar is asked, I will give you a simple diagram. This is pentose sugar. If I say, let me give you this structure. See, this one is first carbon, second, third, and then fourth, and then fifth. So here what happens, nitrogenous base is going to attach to the first carbon of the pentose sugar, and then phosphate is going to attach to the fifth carbon. In detail explanation if you want, I will give you in detail also. That's here like this. This is the best diagram I can say. This is pentose sugar, isn't it? This pentose sugar, first carbon, second, sorry, this one is first, second, third, fourth, and then fifth. So for the first carbon what happens, there will be the attachment of the nitrogenous base. What are the nitrogenous base? Either may be purines or else pyrimidines. Purines are pyrimidines, isn't it? Purines how many? Pure as gold, that is A and G. Pyrimidines CTU. So these or any one can be attached here at the first carbon. If I ask where the phosphate group is going to attach, phosphate group is going to attach at the fifth carbon, and OH group is going to attach to the third carbon of the pentose sugar. OH group is going to attach at the third carbon, whereas phosphate group is going to attach at the fifth carbon, and then nitrogenous base is going to attach at the first carbon. I hope this is clear. So 11th question. Nitrogenous base is linked to pentose sugar by which bond? Nitrogenous base is linked to pentose sugar by which bond? Means what he is asking, this is pentose sugar, isn't it? And here if I say there is a nitrogenous base, what is this bond he is asking? Okay. Option A, N-glycosidic bond, B, phosphoester bond, C, phosphodiester bond, and then D, peptide bond. Here the right option is option A, N-glycosidic bond. This bond is said to be what? N-glycosidic bond. Say this is I drawn like this. This is simplified diagram, and diagrammatically if I ask, see this is pentose sugar, isn't it? And in this pentose sugar what happens, if the nitrogenous base is attaching, that nitrogenous base is attached by a bond which is called what? Beta N glycosidic bond. And the phosphate group is attaching to the fifth carbon of the nitrogenous base, and that attachment is said to be what? Phosphoester bond. Phosphoester bond. And what about phosphodiester bond? When one more nucleotide, means there will be a bond between two nucleotides, then it is called what? Phosphodiester bond. Okay, so nitrogenous base attached at the first carbon. So here the right option is N-glycosidic bond. N-glycosidic bond is bond between pentose sugar and nitrogenous base. Phosphodiester bond is bond between pentose sugar and then phosphate group, this one. Okay, and N-glycosidic is this one. If I say phosphodiester, what is phosphodiester? Likewise, if I am drawing one more nucleotide, let me draw here. This is one pentose sugar binding with nitrogenous base with N-glycosidic, and there will be a phosphate group, isn't it? This is one nucleotide. If I draw one more nucleotide with another base pair, this is phosphate group, isn't it? So here, if the bond between the third carbon and then phosphate is happened, then that bond is said to be what? Phosphodiester bond. This bond is phosphate, the bond between two adjacent nucleotide. I hope this is clear to everyone. 12th one, phosphate group is linked to which carbon of the pentose sugar? Which carbon of the pentose sugar? Fifth carbon, that is option D is right. See here, I will zoom. First carbon, second carbon, third carbon, fourth carbon, here will be somewhat fifth carbon. So this is the pentose sugar. For the first carbon, what happens? Nitrogenous base will be attached, either purines or else pyrimidine. In DNA, pyrimidine will be cytosine and thymine. In RNA, thymine is replaced by what? Uracil. Okay, so here second carbon. At the third carbon, OH group is going to be attached, and then for the fifth carbon, phosphate group is going to be attached. The phosphoester bond between the 5' carbon of the sugar and phosphate group from the 5' end of the nucleotide. This is the free end, isn't it? So phosphate group is attaching at the 5'. Which bond? Phosphoester bond. If I ask the bond name, phosphodiester bond, it will be between the 3' OH of this sugar and 5' OH of the next sugar, that is going to be joining next sugar. So joins the nucleotide to form the DNA or RNA backbone. Phosphate at 5, chain through 3' to 5'. Base and then sugar. What is the bond? N-glycosidic. Sugar and phosphate, phosphoester. Sugar and sugar, phosphodiester. This one is one sugar, and next coming one more sugar will be there. No? The bond between that two sugar is called what? Phosphodiester. This is a quick trick you can remember very quickly. So next question, that is question number 13. Identify the free ends of given polynucleotide chain. This is one end and this one is two end. See here, this phosphate is present, isn't it? If phosphate is free, then it will be called as 5'. If OH is free, what it is free means? OH is free, no, nothing is joined here, and that end is said to be what? 3' end. So 1 is 5' phosphate end, and then 3' is hydroxyl end. So here the right option is option C. 1 is 5 dash end, and then 3 is 3 dash end is hydroxyl. Why it is called 3 dash, why it is 5 dash? Because phosphate group is attaching to the fifth carbon of the pentose sugar, and it is free. That's what it is called 5 dash. And whereas why it is called as 3 dash? Because OH is attaching to the third carbon and it is free. That's what it is called, what? 3 dash hydroxyl. I hope this is clear. That's good. Next, 14th one, backbone of polynucleotide chain is formed due to A. Sugar and N base, B. Sugar and phosphate, C. Phosphate and N base, D. All of this. Here the right option is option B, sugar and phosphate. If I draw polynucleotide, poly in the sense many, nucleotide means you know that this is one nucleotide, that is pentose sugar, and then base here, and here what will be? There will be a phosphate group. This is one nucleotide. If I draw many nucleotides, then it is called what? Poly. Poly in the sense many. One more nitrogenous base here, and there will be one more phosphate here. 2 nucleotides I drawn already, and one more nucleotide let me draw. Nitrogenous base, and then this is rough diagram, okay na? This is N-glycosidic bond, and this is phosphoester bond. If the bond happens between 2 nucleotides among the free phosphate group and then OH, then it is called what? Phosphodiester bond. Here also same. Now this can be called as what? Polynucleotide. So here backbone of polynucleotide is made up of what? Phosphate and then sugar, where nitrogenous bases are inverts. So this is the backbone. If I say backbone is made up of what? Only phosphate group and then pentose sugar. That's what the right option is, option B. 15th one, which is correct about thymine and uracil? A. Uracil is 5-methylthymine. Uracil is 5-methylthymine. Whereas B, thymine is 5-methyluracil, and then C, uracil is 5-ethylthymine, and then D, thymine in 5-methyluracil. Thymine in 5-methyluracil. So here the right option is option B, thymine is 5-methyluracil. Next question, that is question number 16. DNA is A. acidic and positively charged, B. basic and positively charged, C. acidic and negatively charged, and then D. basic and negatively charged. So here the right option is option C. It is acidic and then negatively charged. Why it is said to be acidic? Because DNA contains phosphate group, isn't it? Just now I explained about that. In its sugar, that is whatever the sugar and then phosphate backbone is there, no? These phosphate group have a hydroxyl group, that is it is indicating OH, phosphate group OH, isn't it? That can be denoted a proton, which is a defining characteristic of an acid. Whatever the backbone, the sugar phosphate backbone is there, no, that phosphate group has what? Hydroxyl group. Hydroxyl group. This hydroxyl group is denoting as a proton. So which is defining characteristic of what? Acidic. Why it is called as negatively charged? Why the DNA is called as negatively charged? When those phosphate group donate their hydrogen ions, they are left with the negative charge. When they are going to donate, or they are donating hydrogen ions, so this gives the entire DNA molecule a net negative charge. Okay na? That's why DNA fragments move towards positively electrodes, because it is negatively charged. Next, 17th one. Name of DNA as nuclein was given by. The name of the DNA as a nuclein was given by whom? A. Francis Crick, B. Erwin Chargaff, D. Frederick Mischer, and then D. Rosalind Franklin. Here the right option is option C, Mischer. Next question, that is question number 18. Double helix for structure of DNA model was proposed by. Double helix for structure of the DNA model was proposed by A. Wilkins and Franklin based on their X-ray diffraction data. B. Watson and Crick based on their X-ray diffraction data. C. Chargaff based on their X-ray diffraction data. And then D. None of this. Here the James Watson and Francis Crick proposed the double helix model. They proposed double helix model, which is heavily based on what? X-ray diffraction method. X-ray diffraction method. So the right option is option B. They discovered in the year 1953. Next, the proposition of base pairing between two strands of polynucleotide chain in double helix model of DNA was based on observation of Morris Wilkins, Rosalind Franklin, Erwin Chargaff, and then both A and C. Already answer is revealed, that is C. Here Chargaff rule, what Chargaff rule says? Always adenine pairs with thymine, and then guanine pairs with cytosine. Okay, adenine and thymine are same, and then guanine and cytosine are same. Means purine pairs with pyrimidine. What is this pairing means? Means if I say this is the pentose sugar and there will be a nitrogenous base, I said, isn't it? And here there will be a phosphate group and one more nucleotide. If I am drawing one more phosphate group, and here is the nitrogenous base. If I say the nitrogenous base, if I am taking a purine here, that is A, and then here I will take G, okay, and the pair, that is phosphodiester bond. To the opposite, you know DNA is double helix, isn't it? This is only single nucleotide chain I am drawn. To this one, oppositely, there will be again pentose sugar, which will be anti-parallel. Anti-parallel means the phase will be opposite. I hope you people are getting. And nitrogenous base will be inwards. And one more pentose sugar here. And here it is phosphate. And here is phosphate, and the bond between them somewhat okay. So here A always pairs with what? T, and G always pairs with C, vice versa. Okay, A always pairs with T, and then G always pairs with what? Cytosine. here what happens, A always pairs with T and then G always pairs with C. Through which bond? Through hydrogen bond, to hold the two strands together. Okay na? This is hydrogen bond and this is also hydrogen bond. So this is what actually the structure looks like. So adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. Okay, this is one polynucleotide chain and this one is another one polynucleotide chain. They run anti-parallel. They are not running parallelly. Parallel in the sense phase must be same. Why it is called anti-parallel? Because one direction is this one and one more is this one. See here, free is OH. If OH is free, what that end is called as D, 3 dash. If phosphate is free, then it is called what? 5 dash end, isn't it? So let me ask you question, what is the free here? 3 dash is free, so this end is 3 dash. Here phosphate is free, so what it is called? 5 dash end, isn't it? So this is running in one direction and this one is running in one direction. That's what they are called, anti-parallel. Keep in mind, always adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. So this is about the Chargaff rule. Next, 20. The 2 chains of double helix DNA have parallel polarity, anti-parallel polarity, no polarity, depends on the organism. Just now I explained very detail about that, that is anti-parallel polarity. See here, if you see, let me zoom that one. See here, phosphate is free, so this end is called what? 5 dash end. Here OH is free, that is hydroxyl is free, so this is called as 3 dash. And if you see another end of the blue strand, another end will be having 3 dash, and if you see the green one, it will be having 5 dash. So they are blue is running in this way and then green is running in this way. So what is the direction? If I say 5 dash to 3 dash and 3 dash to 5 dash, that's what it is called, anti-parallel. 21. The bases in 2 strands of DNA are paired through, I just said, hydrogen bond, peptide bond, glycosidic bond and sulfide bond. Here the right option is hydrogen bond. What are the bases? 2 strands of bases, that is nitrogenous base he is asking. If A is there, A is pairing with what? T. With how many hydrogen bonds? 2. If G is there, it is pairing with C. With how many hydrogen bonds? 3 hydrogens. If I ask C is there, to whom it is going to bond? It is going to bond with the G. If I am saying T is there, it is going to bond with the adenine with the 2 hydrogen bonds. Vice versa I said, no, this is the vice versa. Next one, 22. Which of the following is true about base pairing in DNA? Base pairing in DNA. A. Adenine forms 2 hydrogen bond with guanine. Adenine forms 3 hydrogen bond with guanine. No. Adenine forms 2 hydrogen bond with thymine. Yes, this one is correct. Just now I said, isn't it? So the right option is option C. A always pairs with T with 2 hydrogen bonds. AT to GC3. A, in between the adenine and thymine there will be 2 hydrogen bonds. In between C and then G and C, there will be 3 hydrogen bonds. Trick to remember, GC has a great connection. Great connection. Why great connection? Because it has a 3 hydrogen bonds, isn't it? G and then C. Next, 23. Which of the following is true about base pairing in DNA? Question is same, but the options are different here. Don't get confused. A. Guanine forms 2 hydrogen bond with cytosine. No. Guanine forms 3 hydrogen bond with cytosine. Yes. So here the right option is option B. Next, 24. Uniform distance between 2 strands of helix is due to uniform distance. A. Double and then triple bond formed between the base pairs. B. Sugar and then phosphate backbone. C. Purine and pyrimidine base pairing. D. None of this. Here the right option is C, that is purine and pyrimidine base pairing, which leads to uniform distance between the 2 strands of the helix. 25. How many of the following statement about double helix structure of the DNA is correct? How many among the following statement about the double helix structure of the DNA is correct? So here the first one is two chains are coiled in right-handed fashion. 2 chains are coiled in right-handed fashion. Yes, of course, this is right. The 2 polynucleotide chains are coiled in right-handed fashion, that is clockwise direction. Like this they are going to coil. Second one, pitch of helix is 3.6 nanometer. This is incorrect. In the DNA, that is, if I say standard DNA, that is bDNA model, the pitch is 3.4 nanometer, not the 3.6 nanometer. Next, then third, there are roughly, this is not correct because there is a 3.4, not the 3.6. So this one is wrong. There are roughly 10 base pairs in each turn. Yes, exactly correct. Plane of one base pair stacks over the other. This one is also correct. So here 1, 2, 3 are correct. What he is asking? Correct one. So 3 is correct. So option C is correct. Next question. 26th one, that is pitch of helix in double helix DNA is. Pitch of helix in double helix DNA is 3.6. No. 3.4. Yes, just now I said about that, isn't it? What is pitch actually? Pitch is one turn. If I say this is one helix and this one is one more helix, from this to this, whatever the turn is there, no, that is said to be what? One helix, one turn. One turn, okay. This one will be 3.4 nanometer. 3.4 nanometer, or else you can also write it as what? 34 angstrom. The length from this to this is called what? One helix. Okay, or else pitch of helix is said to be 3.4 nanometer or else 34 angstrom, and it will be having roughly 10 base pair. This is one base pair, 2, 3, 4, 5, 6, 7, 8, 9, 10, isn't it? So as it is having 10, the distance between the base pair, if I ask what is the distance, this one, if I ask, what you'll do? So total how much it is? 34. How many base pairs are there? 10. So I will be getting this distance. How much? 3.4 angstrom or else 0.34 nanometer. Okay na? Let me write here this one, this distance 0.34 nanometer. Okay, don't get confused. I hope this is clear. From this to this, one helix distance is 34 angstrom or else 3.4 nanometer. If I ask the distance between two base pairs, this is one base pair and this one is one more base pair. The distance between two base pair is how much? 0.34 nanometer. So here he is asking pitch of helix. So this is the pitch he is asking. So the distance is 3.4 nanometer. Next, 27th one. Central dogma in molecular biology was proposed by A. Crick, B. Watson, C. Misher and then D. Chargaff. Here the right option is A. Crick. Next, 28. Identify the correct labels. This is 1 and then this one is 2, this one is 3. See, if DNA is giving DNA, then it is called replication. From the DNA, RNA is forming, especially mRNA, then it is called transcription, and mRNA is giving protein, means then it is called translation. Okay, replication, transcription, translation. So here the right option is option B. From the DNA to DNA, replication, DNA to RNA, transcription, RNA to protein, translation. DRP, direction of life. DNA is giving RNA, RNA is giving protein. So now next question from the concept packaging of DNA helix. This is one of the best question I can say. That is question number 29. If the length of the E. coli is a bacteria, length of the DNA is given, that is 1.36 mm. Calculate the number of base pairs. Number of base pair he is asking in E. coli. And what is given? Distance between the consecutive base pair, that is 0.34 into 10 to the power of minus 9 m. So here the option, before the option giving, let me give you a very short trick. Okay, see how you can calculate this one. This much long length calculation is also there and a very short one is also there. Let me explain first. Okay, see, whatever he is given, length of the DNA is given. What is the length of the DNA? 1.36 mm. It is also equal to 1.36 into 10 to the power of minus 3 m. Distance between base pairs. If the length of the DNA is this much, what is the distance between base pair? Base pair in the sense, in a small area there will be a bases. The distance among them is given how much? 0.34 into 10 to the power of minus 9 m. What formula we are going to use? We want what number of base pair we want, isn't it? So here number of base pair is equals to total length and then distance between the base pair. Distance between the base pair. What is the total length? 1.36 into 10 to the power of minus 3 divided by 0.34 into 10 to the power of minus 9. If I divide the numbers, I will get the 4, isn't it? I got 4. 4 is there in all the answers. What about this? Handle the powers. If I handle the powers, 10 to the power of minus 3 divided by 10 to the power of minus 9 will be giving how much? 10 to the power of 10 to the power of 6. So where is 10 to the power of 6? It is here. This much lengthy process is not necessary. So the final answer will be 4 into the 10 to the power of 6. Simply just the length of the DNA is given. This is the simple trick, super trick I can say. The length of the DNA is 1.36 and then divided by 0.34. Just add to this 10 to the power of 6. So answer is 4 into 10 to the power of 6. I hope you people are understood. Next, 30th question. In E. coli, DNA is scattered throughout the cell. In E. coli, DNA is scattered throughout the cell, and then reason. In E. coli, there is no defined nucleus. So here the right option is A itself, because assertion and reason are correct and reason is correct explanation of assertion. Yes, of course, in E. coli DNA is scattered throughout the cell because it is a prokaryote, and in E. coli there is no different nucleus because of the prokaryotic characteristic. There is no defined nucleus. So option A is correct here. This one is wrong. Make it as A. Next question. Histones are, 31st question. Histones are A. Positive and acidic in eukaryotes, positive and acidic in prokaryotes, positive and basic in eukaryotes, and then D. Positive and basic in prokaryotes. Here the right option is the charge is positive and then they are basic in eukaryotes. So the right option is option C. Why it is positive and then why it is basic in nature means histones are very rich. They are very rich in lysine and then arginine. They are the amino acids. They are very rich in what? Lysine and arginine. So due to the presence of lysine and arginine, what happens, which give the positive charge side chains. This positive charge is crucial because it allows the histones to bind tightly to the DNA. See, as you know, just imagine this is the histone and this is the DNA. DNA is negatively charged, and if the histone is positively charged, what happens? The wrapping will be very tight for the packaging, isn't it? That's what histones, due to the presence of lysine and arginine, histone has positively charged. And why it is basic? Because of the high concentration of basic amino acid, that is lysine and arginine. Both are basic amino acids. Next one, 32th question, assertion and reasoning type of question here. Histones are positively charged. Reason, histones are rich in basic amino acid residue lysine and arginine. Both are correct and this is correct explanation of this. Just now I explain. I hope there is no necessary to explain once again. So A is correct. Next, 33. Histones are organized into. Histones are organized into hexamer, octamer, tetramer and dimer. No, they are organized into octamer. If I say this is the histone 1, 2, 3, for simplification I am drawing this one. H2A, H2B, H3 and H4 there will be. H2A, H2B, H3 and then H4 there will be. If I say this is H2A and H2B, H3, H4, this can be like, okay, this is 4, same like that there will be 2, 2, So they become 4, 8. Okay, H2A will be 2, H2B2 will be 2, H3 will be 2, and H4 will be 2. That's what it is called, what? Octamer. Histone are organized into octamers from 1, 2, 3, 4, 5, 6, 7, 8. Next question, that is question number 34. A typical nucleosome contains dash base pair of DNA has. A typical nucleosome. What is nucleosome actually? It is a basic structural unit of DNA packaging in eukaryotes. What is basic structural unit? See, the DNA is too long, isn't it? It's almost about 2 meter long. That 2 meter long DNA, how it is going to be possibly present in a small tiny area which is called as nucleus, isn't it? This much long DNA is situated in this small tiny area which is called as nucleus by packaging method. Means there will be a high coiling. Due to that, what happens, there is a possibility of being there. If I say which is the basic unit of the packaging, basic structural unit of packaging means nucleosome packaging, which is having the histone and the DNA which is wrapping around it. So from this to this, whatever the DNA is packaging, if I take that one from this to this, if I take the length, how much base pair it is having, he is asking. What is the base pairs? It is having 200 base pairs. Okay, and then this is nucleosome. Nucleosome core is made up of histones, you people already know, and that histone is wrapped by the DNA. Next, 35. Repeating unit of chromatin. A are nucleosomes. B are seen as beads on a string under electron microscopes. C are packed to form a fibers. D all of this. Here the right option is all of this. Next, 36th. This figure shows beads on a string, correct? This one is beads 1, 2, 3, 4. These all are beads of a string. Here beads are nucleosome and then string is what? DNA. Next, see, this is what actually the nucleosome structure, and this one is the core. Core is having what? Histone core, which is a octamer, and this is the DNA, and this one is one more histone, which is H1 we used to call. This is H1, this is DNA, and this one is nucleosome or else core of histone molecule. Here what will be? There will be H3, 1H3, H4, H2A and H2B there will be. Okay na? So here what? 2 is DNA. So either it may be A or D. 1 is H1. So option D is correct here. Next, 38. Chromosomes are connected, chromatin fibers present. Chromosomes are connected, chromatin fibers are present. Option A, at all time in cell. B, only during cell division formed at the prophase. C, only during cell division formed at metaphase. D, only during cell division formed at interphase. And here the correct option is only during cell division formed at prophase. Make it as B, it's not C, it's B. Next, 39th one. In a typical nucleus, euchromatin and heterochromatin are present. Choose the correct set of characters of heterochromatin. Heterochromatin's correct characteristic you must choose. Here heterochromatin, he is asking about heterochromatin. They are loosely packed? No, they are densely packed. There will be tight packaging. Due to that, what happens? They show dark stain, not the light stain. They are inactive chromatin because of the densely packaging. Which one? Heterochromatin. So they are not the active one, they are inactive. So 2, 4 and then 5, 2, 4 and 5, that is D. Option D is correct. This is true about the heterochromatin. Heterochromatin is densely packed, darkly stained and inactive chromatin. If I show you the diagram, see, this is the very densely packed one and this is very loosely packed. That's what it is called, heterochromatin, and then this is called euchromatin. This is the nucleus. Inside the nucleus there is a DNA. That DNA portion is zoomed here. Heterochromatin is highly condensed, darkly stained, it is inactive and then not transcribed. That's what it is called, inactive. Found near the nuclear membrane. They are found near the nuclear membrane. And if I talk about euchromatin, they are loosely packed, lightly stained, very active and transcribed, and it is distributed throughout the nucleus. So next question, it is about 40th question. Choose the correct set of characters for euchromatin. Number 1, loosely packed, yes. Number 2, densely packed, no, it is for hetero. 3, lightly stained, yes. Darkly stained, no. Inactive, no. Active, yes. 1, 3 and then 6. 1, 3 and 6. So right option is option C here. 41, the search of genetic material. From here you will get many experimental questions. This is one of the favorite topic for the NEET. You will get compulsorily one question from the experimental kind of questions. First experiment, that is the transforming principle. First one, 41 question. Griffith experiments were conducted in. Griffith experiment were conducted in A, 1928, B, 1958, C, 1978 and then D, 1968. Here the right option is option A, 1928. In the 1928, British bacteriologist, he is a bacteriologist actually, Frederick Griffith conducted a series of experiments using Streptococcus pneumoniae, a bacteria that causes what? Pneumonia. And he is very famous for the transforming principle. 42. The experiment of Griffith was performed in A, Diplococcus pneumoniae, that is a bacteria. B, Haemophilus influenzae, that is fungi. C, Streptococcus pneumoniae, that is fungi, and then D, none of this. Here the right option is option A. You will get confused. Ma'am, just now you said that Griffith worked on Streptococcus pneumoniae. Of course, he worked on Streptococcus pneumoniae, but it is not a fungi, it's a bacteria. And why we choose the option first? Because Diplococcus pneumonia is nothing but an older name of Streptococcus pneumoniae. Okay na? And it is a bacteria. So this is more correct than this, because Streptococcus pneumonia is not a fungi. Okay na? Next, 43. 43, match the following type of question. Here, one, that is strains of that bacteria, Streptococcus pneumonia bacteria is given. R strain, that is rough strain. R strain has a rough colonies, S strain has a smooth colonies. S strain has a smooth colonies, and then whereas R strain has a rough colonies. Smooth is showing you what? Mucus coat. There will be a mucus coat in the smooth, and then rough don't have what? Mucus coat. So 2 is matching for what? A, and then 1 is matching for B, and then 2. So here B 1 and 2 is wrong. It is not there, it's not there, it's not there. Let me write 2 is matching for A and then 1. So we have C. Which strain of microbe used Griffith is virulent? Among the S and then R strain, Griffith used 2 strains. Okay, let me explain that one. He used what? 2 strains. One is S, one is R. Which one is virulent means S is virulent, and then R is non-virulent. Why S is virulent? Because it is having the mucus coat. Why RN is not virulent? Because it was not having that one. So here the right option is option A. Next, 45. Griffith observed that the mice died surprisingly. The following combination of strains was used, which was unusual. Which was unusual? He is asking. A, S strain heat killed. B, heat killed S strain. C, heat killed R strain plus live S strain. And then D, heat killed S strain plus live R strain. See here what happens. The right option is option D. When you are heating the S strain means the virulent is already dead, isn't it? But the dead virulent is when it is mixing with the live RNA. Unexpectedly what happens? The mice was died. When the Griffith experimented started, he first what he did? R strain he heated and he killed and then injected to the mice. Mice was alive. Then next, heat killed S strain again. If he is, see, let me explain here separately. Firstly, he used S strain and injected to the mice, and then mice was died due to pneumonia. Next what happens? He used rough strain and injected to mice, and then mice was alive because it was non-virulent. Again what he did? He heat killed rough strain and the normal S strain, and he injected to mice, and then mice was died. Next time what he did? He heat killed S strain and it is mixed with live R strain, and then the mice was what? Mice was dead. What he was expecting? He was expecting mice might be alive because it is heat killed already. If R strain is heat killed, what is the possibility? Heat killed means it must be alive, and whereas the live R always R is what? Alive. So it must be also alive. He was expecting mice must be alive, but the result was what? The mice was dead. So this is the unusual expectation of the Griffith when heat killed S strain is mixed with the live R strain. So here the dead gives life. Dead gives life means smooth strain which is virulent, which was dead, gives life to the R strain. Why it is happened means whatever the DNA present in S strain is there, no, that is transformed to the live R, and then it is started to form a mucus coat around the rough strain. So dead is yes, makes are dangerous. Who makes are dangerous? Yes, makes are dangerous. This is what actually the transforming principle explains. Next, 46. In Griffith experiment, R strain transformed to S strain and become virulent. R strain transformed to S strain and lost virulence. S strain transformed to R strain and become virulent. S strain transformed to R strain and lost virulent. So here the right option is A itself. R strain transformed to S strain and become what? Virulent. 47. Griffith claimed that some proteins was transferred among the bacteria. Some DNA was transferred among the bacteria. Some carbohydrate was transferred among the bacteria. None of this. Here the right option is none of this. Why? Because he said, of course he said the transforming principle. He didn't use whether it is protein, whether it is DNA, whether it is carbon dioxide or carbohydrate. He just said that something is transferring. He didn't mention it. Okay, that's what none of this is. Option is correct. Next, biochemical characterization of the transforming principle. 48. Prior to work of Avery, MacLeod and McCarthy, genetic material was thought to be protein. Before the invention, that is genetic material of the DNA, we thought that protein was the genetic material. Next, 49. Avery, MacLeod and McCarthy discovered that option A, DNA caused transformation, B, RNA caused transformation, C, protein caused transformation, and then D, lipid caused the transformation. So here the right option is A, that is DNA caused the transformation. See, they continued the Griffith experiment, isn't it? Whatever the Griffith said, no, he didn't mention which one is going to be transformed. But finally, Avery, MacLeod and then McCarthy discovered that DNA is the transforming material which is going to be transferring. DNA was the substance which is responsible for the transformation of the non-virulent R strain to the virulent S1. Okay, na? Next, 50th question. Which enzyme inhibited the transformation? Which enzyme inhibited the transformation? That is option A, protease, B, RNase, C, DNase, and then D, all. Here the right option is option C, DNase. What actually this means? Inhibited the transformation. See, if the protease is added to a test tube for the conduction of transformation experiment or else transforming principle, protease, what happens, this enzyme digests the protein. When it is added to the transformation, what happens, transformation still occurs, meaning what, protein was not the genetic material, isn't it? If I am using RNase enzyme digests what, RNA. When it is added, transformation still occurs, meaning what, RNA was not the genetic material. If I am destroying the DNA, that is DNA is added, means what happened, DNA is going to be destroyed. If DNA is added, then it is transformed, there will be inhibited, there will be no transformation was inhibited. That's what the enzyme which is inhibited the transformation is DNase. This what it is proved that DNA was the substance responsible for the transforming. Next, the genetic material is DNA from the portion. 51. Unequivocal proof that DNA is genetic material came from experiments of A. Avery, MacLeod and McCarthy, B. Hershey and Chase, C. De Vries and Correns and T.S. Charmak, D. Sutton and Bouvier. Here the right option is option B, Hershey and Chase experiment, which is unequivocal proof that DNA itself is a genetic material. What it is called? It is also called as blender experiment, where DNA is isotoped by phosphorus, that is P32, and whereas protein, that is sulfur, is isotoped with S35. How you will remember? DNA P, protein S. P enters, S stays. Okay. What it is P enters, S stays, I will explain in later on. There is a diagram in upcoming slides. I will explain about that. 52. The scientists of Q11 worked with. The scientists of Q11 worked with a virus, a bacteria, a fungi, a nematode. Here the right option is option A, a virus. Here Q11 refers to what? Hershey and Chase work. Okay na? Next one, 53. In the experiment performed for providing DNA as genetic material, the bacteriophage were grown on medium containing. They were grown on the medium containing radioactive sulfur, radioactive nitrogens, radioactive phosphorus. So the experiment performed for the providing DNA as genetic material, the bacteriophage were grown on a medium containing radioactive sulfur and then radioactive phosphorus. So the right option is more than one option is correct. 54. The bacteriophage growing in presence of radioactive phosphorus dash contains radioactive dash. First one is what and then contain radioactive. First one is P32 and contained radioactive DNA. Here he is asking phosphorus. So for the phosphorus, radioactive P32. Next, bacteriophage grown on a radioactive sulfur dash contained radioactive dash DNA. 2 is DNA compulsorily. What is yes? Yes must be what? Yes 35. Here only I said no. See, yes 35 and then P32. So here the right option is yes 35 DNA. C, not the D1. C option is correct. Next, 56. The bacteria involved in Hershey and Chase experiment of 1952 was bacteriophage, E. coli, Streptococcus pneumoniae and then Botulinum. So here the right option is E. coli. 57. Bacteria infected with virus that showed radioactivity had radioactive DNA S32, S35, radioactive DNA P32, radioactive DNA P35. Here the right option is radioactive DNA P32. Whatever the experiment is done by Hershey and Chase, no, bacteria which was infected with virus that showed radioactive had what? Radioactive DNA P32. Next question, that is question number 58. See, this one is infection. This is what actually Hershey and Chase experiment diagram. First stage or first step is infection. next is blending, next is centrifugation. So with respect to the correct option is option B. What happens here, this is bacteriophage, a virus which infects the bacteria. Okay, this bacteria is E. coli. And here what happens, radioactive S35 is labeled. Radioactive S35 means only protein is radio labeled. Only here protein is labeled, protein code. Here radioactive P32 means DNA is labeled here. DNA is radioactive. Here only outer coat is radioactive. If coat is radioactive and then the inner DNA is radioactive, both the different bacteria were infected to the E. coli. What happens here, normal DNA is entered and that DNA is replicated, and then at the last, after the centrifugation, there is no radioactive detected in cells. Why? Because radioactive protein is outside only, isn't it? Radioactive detected in supernatant, that is in supernatant it is detected. Next, and here if the radio label is for the DNA, that is the material, genetic material, if this bacteriophage is infecting to the bacteria, its DNA is entering inside. Okay, after entering, DNA is going to be multiplied and that DNA is going to be shifted to its next generation, that is doctor bacteriophages. So after centrifugation, what happened, radioactive detected in cells, that too specifically P35. Why? Because the transferred was what, DNA, not the protein. So no radioactivity detected in supernatant. Next, properties of genetic material, that is DNA versus RNA. 59. RNA is genetic material in A. TMV, B. QB bacteriophage, C. both A and B, D. none of this. Here the right option is C, both A and B. 60. Properties of genetic material includes A. stable, B. mutable, C. replicable, and D. all of the above. C. Here the right option is D, all of the above. A genetic material must include stability, mutation it must be having, and then replication. 61. A stability as a property of genetic material was very evident in Griffith's transforming principle. Yeah, it's true. Heat can kill the bacteria and 11 completely destroys the properties of genetic material. Heat can kill the bacteria and completely, and it's not 11, and completely destroy the properties of genetic material. It's what? It's false actually. Because while heat can indeed kill the bacteria by denaturing their proteins and disrupting the cellular process, but it doesn't completely destroy the properties of what? Genetic material. So this one is wrong. So the right option will be C, because A is true, B is false. 62. Which is more structurally and chemically stable? Which one is more structurally and chemically stable among the following? Option A, DNA, option B, RNA, C, protein, and then D, all. Here the chemically stable one is DNA. So the right option is A. DNA is chemically stable, structurally stable, and then it is base composition. DNA uses thymine instead of uracil, whereas uracil is found in RNA, and it has genetic storage also. Because of the high stability only, genetic storage is very safe in the DNA. Next, RNA viruses show 63. RNA viruses show A. Less mutation, B. Faster evolution, C. Slower evolution, and then D. More than one option is correct. Here, when we talk about with respect to the RNA virus, they are going to evolve very fast. So B option is right here. 64. DNA is preferred by nature over RNA for DNA is preferred by the nature over RNA is for what? A. Storage of genetic information, B. Transforming of genetic information, C. Expression of genetic information. More than one is right here. The right option is A. Storage of genetic information. See what happens actually. Why DNA is preferred over the RNA? Because while both DNA and RNA can carry genetic information, but DNA has several structural advantages that make it a ideal choice for the long term storage in the most organisms. Why? Because DNA is chemically stable, just now we discussed, and it is double stranded structure, and it has thymine instead of uracil. So next, from the questions from RNA world. 65. Choose the incorrect statement for the RNA. Option A was first genetic material, B acts as catalyst to, and then C is more stable than the DNA. This one is incorrect. DNA is more stable than the RNA and has protein synthesizing machinery built around it. So this one is also right, but the C one is incorrect. So he is asking incorrect statement among the following. Next, 66th. Scheme for replication of DNA was proposed by. The scheme for the replication of DNA was proposed by A. Watson and Crick, B. Michelson and Starr, C. Taylor, and then D. Hershey and Chase. So here what happens? Watson and Crick is right, whereas Michelson and Starr experiment, what they are proven, they are credited with the proving the semi-conservative replication scheme through their experiment, that is N15 and N14 isotope they are used, nitrogen isotope they used. Where about Taylor, Herbert Taylor, you can say, performed similar experiment using radioactive thymidine in plant roots, that is especially Vicia faba. It is also given in NCERT. When it comes about Hershey and Chase experiment, they performed about the blender experiment. Here for the replication of DNA was proposed by whom? Watson and Crick. Next one, that is 67th question. The replication of DNA is A. Conservative, non-conservative, C. Semi-conservative, D. All of these depend on the organism. Here the right option you people already know, that is semi-conservative. Next, the figure shows, this figure shows conservative DNA replication model, semi-conservative DNA replication model, non-conservative DNA replication model, and can't say. Here the right option is option B. See, this is the semi-conservative method. What is semi-conservative method? This is the main parental DNA and the newly forming DNA is forming here, isn't it? So like this, formed daughter DNA will be having one parental strand and then one newly formed. That's what it is called semi-conservative. One is conserved from the parent and one more is newly formed. Next, the experimental proof. 69. The DNA replication model experiment proof was first shown in human cell, B. E. coli, C. plant cell, D. Vicia faba. Here the right option is option B, in E. coli. The semi-conservative model of the DNA replication was first provided by Michelson and Starr in the year 1958, isn't it? And the organism used for that is Escherichia coli. Missels and Starr performed experiment for providing DNA replication scheme in the year 1958. 70th question. So right option is C. Next, 71. The bacteria were grown in medium containing. The bacteria were grown in the medium containing A. 15NH4Cl. 15N is heavy isotope of nitrogen. And then option B. 14NH4Cl. 14N is heavy isotope of nitrogen. 15NH4Cl. 15N is normal isotope. 14NH4Cl. 14N is normal isotope of nitrogen. Here the correct option is option A. 15NH4Cl, whereas 15N is heavy isotope of nitrogen. Next question, that is question number 72. The heavy DNA molecule containing heavy isotope of nitrogen is distinguished from normal DNA by A. UV rays, no. B. Ethidium bromide solution, no. Centrifugation is cesium chloride density gradient. So C is correct here. Next, in 73, in Missels and Starr experiment, first they A. grew bacteria on heavy isotope of nitrogen medium followed by normal one. Next, they grew bacteria on normal isotope of N medium followed by heavy one. They grew bacteria on radioactive N followed by heavy one. Grew bacteria on heavy isotope of N followed by the radioactive one. Here the right option is option A itself. See here, identify the correct label. 1 and then 2 and then 3 is given. This is 1, this is 2 and then this one is 3. This is generation, this is heavy isotope. After semi-conservation, what happens? One parental strand is here and one more is here, and newly formed is in the form of green one. Okay, newly formed will be light because this is heavy, which is often and often it is multiplied in the heavy isotope labeled medium. Once the first generation is come, what happens there is a semi-conservative method. Again, if we grow this E. coli in the next generation, what happens? There will be 4 strands for the parental DNA and the newly formed will be there. And when we centrifugate them, we will find a light band also and then heavy band also. Light band and then hybrid band also. Why the light one? Because the newly formed will be of light. 1 and 3 are light, and then 2 are what? Hybrid. We won't find heavy. We are not finding heavy. Why? Because they are not totally transferring to the next generation. So here first one is light, one is light. A and C both have light. Second one is hybrid, heavy, no. So this is hybrid. So right option is hybrid. 75 question. In Missels and Starr experiment, a bacteria after dividing in 20 minutes had a hybrid DNA. What will be the ratio of hybrid to the light after 80 minutes? After what? 80 minutes he is asking. What is the ratio he is asking here? So total generation, how many? 80 minutes he is given. 20 minutes had a hybrid DNA. 20 minutes had a hybrid DNA means what? Total generation, if I take 80 minutes, bacteria is dividing in 20 minutes. So 80 divided by 20 means how many generation it is divided? 4 generation it is divided. For first gen, what happens? For the generation 1, that is for 20 minutes, there will be formation of 2 molecules. For the next, that is generation 2, there will be 4 molecules, because they are going to divide. And then for the third generation there will be 8 molecules, and for the fourth gen there will be 16 molecules. Determining the hybrid versus light, because DNA is a replication is semi-conservative, the two original heavy N15 strands never disappear, they just get separated and paired with the new light strands, isn't it? So if I take that heavy one, so 16 minus 2, if I do, how much light I will get? 14 lights I will get. So this can be simplified in the form of what? 1:7. But since 2:7 is provided in the option, A will be the correct choice. Here, what is 2 and what is 14? 2 is heavy and then 14 is lighter one. What will be the ratio of hybrid to light? So hybrid, and this one is light. 76. Similar experiment on Vicia faba was conducted by Dash to detect distribution of newly synthesized DNA in chromosomes, that is by Taylor. 77. Experiment on Vicia faba involved use of radioactive uridine, radioactive thymidine, radioactive adenosine, radioactive cytidine. Here the right option is radioactive thymidine. Here in the Vicia faba, radioactive thymidine was used. Machinery and then enzymes for the replication. 78th question. The main enzyme of replication is A. RNA dependent RNA polymerase, B. RNA dependent DNA polymerase, C. DNA dependent DNA polymerase, D. DNA dependent DNA polymerase. Here the right option is C. DNA dependent DNA polymerase. For the replication, DNA dependent DNA polymerase is going to used. Next, choose the correct statement with regard with the efficiency of the DNA polymerase. Option A. 4.6 into 10 to the power of 6 base pair of E. coli replicate within the 46 minutes. Correct. The average rate of polymerization of the DNA polymerase has to be approximately 2000 base pair per minute. This is also correct. The polymerization accuracy is very high and very fast. Yes, exactly. So the right option is option D. All of this is right. So this is not this C, this is D. Next, 80. What is function of deoxyribonucleoside triphosphate? What is the function of deoxyribonucleoside triphosphate? Here it acts as a substrate. B. Provide energy for the polymerization. C. A and B both. It is product formed after the polymerization. Here the right option is deoxyribonucleotide triphosphate. Here the right option is option C actually, because it acts as a substrate also and provide energy for the polymerization. They carry 3 phosphate group. No, what actually the deoxyribonucleotide triphosphate means? DATP DGTP. Here triphosphate is there, no? They provide energy and they act as a substrate also for the polymerization. So here the option C is correct. Please make a correction here. Sorry for the inconvenience. 81. Assertion: the two strands of DNA cannot be separated in their length. The two strands of the DNA cannot be separated in their length. Yes, it is true. Separation require very high energy, and it is also true. So the right option is option A. 82. Polymerization by DNA polymerase is in the direction, always it will be in the direction 5' to 3' direction. So here the right option is option B. 83. The template of replication fork with polarity 5' to 3' is dash, while 3' to 5' is dash. 5' to 3' is discontinuous and then 3' to 5' is continuous. So here the right option is option C. Always direction of synthesis of the DNA polymerase only synthesize a new DNA strand in 5' to 3' direction, isn't it? 5' to 3' direction. If 5' to 3' direction is there, means it adds new nucleotide only in the 3' ends growing strand. That's what this will be the continuous one and then this will be discontinuous one. So the next question, that is question number 84. DNA ligase act on 5' to 3' template strand, 3' to 5' template strand. C. Both A and B. D. Ligate RNA with vector of 3' to 5' polarity. Here the right option is C. Both A and B. That is from the 5' to 3' template strand, 3' to 5' template. You know DNA ligase is what? It's a enzyme which is helpful for the attaching of the fragments, isn't it? It's so DNA repair. For the DNA repair, DNA ligase acts on the both the strands during the DNA repair processes like nucleotide excision repair or else base excision repair. To seal the nicks regarding nicks means there will be small fragments. To join those small fragments, it acts both on them. Next, 85. The replication is eukaryotes takes place in. The replication in eukaryotes, not is, it's in. The replication in eukaryotes takes place option A, M phase, B, G1 phase, C, S phase, and then D, G2 phase. Always keep in mind replication happens in S phase, that is synthetic phase. Polyploidy resulted by a failure in cell division after DNA replication. Next, B, a failure in DNA replication after cell division. C, a failure in cell division before DNA replication. And then D, both A and C. Here the right option is option A, a failure in cell division after DNA replication. Next question, that is question number 87. The diagram is given here. A, B, C and then D, C and then D. A is template strand and then B is newly synthesized. C is discontinuous, that is Okazaki fragments we can say, and then D is continuous one. So A is template strand, B is newly correct labeled A, B, C, D. 1, 2, 3 all are correctly labeled only. So C is right. 88 from the transcription. Transcription is Option A, the process of copying genetic material information from both strands of DNA into the RNA. B, the process of copying genetic information from one strand of DNA into the RNA. This one is correct. Replication is different. Replication is both the strands are going to use there for the replication. But for the transcription, what happens? There will be only use of one strand of the DNA. Next, 89. In transcription, adenosine binds with A. Thymine, B. uracil, C. cytosine, D. A and B both. Here the right option is uracil, because when the DNA is going to be transcribed, if I say this is the DNA, if this is going to be transcribed, this is going to give you the mRNA. So if the DNA is having A, then on the mRNA you will get U, not the T, because RNA won't have thymine. It has, instead of thymine, it has uracil. Next, 90th one. Why both the strands of DNA are not copied during the transcription? Why both the strands of DNA are not copied? A, if both the strands act as template, they would code for RNA molecule with same sequence. B, RNA formed by transcription of both strands, when code for protein, the sequence of the amino acid in protein are same. And then C, the two RNA molecules, if produced simultaneous, would be complementary to each other. So here the right option is C, they would be complementary to each other. 91. Translation of RNA would be prevented if translation of the RNA would be prevented if RNA is single stranded. B, RNA is double stranded. C, RNA is produced by both the strands of DNA. And then D, B and C. So here the right option is option D, B and C. If RNA is double stranded, it is prevented to translate, and then if the RNA is produced by both the strands of the DNA, then also it is going to be prevented. Next, transcription unit. 92. Transcription unit primarily consists of one region, two regions, three regions, and none of this. Here the right option is three regions. 93. Transcription primarily required A, RNA dependent RNA polymerase, B, DNA dependent RNA polymerase, C, DNA dependent DNA pol, D, RNA dependent DNA pol. Here the right option is option B, DNA dependent RNA pol, because in the transcription, depending upon the DNA, we are synthesizing what? RNA. That's what DNA dependent RNA pol we required. The template strands of transcription unit is, the template strand of transcription unit is A, 5' to 3' strand of DNA, and then B, 3' to 5' strand of the DNA, C, site of catalysis of enzyme required for the transcription, and then site of D, both B and C. So here the right option is D. Next question, that is question number 95. What is coding strand of given template strand, that is 3' CATG CA and 5'. If I write the complementary, for A there will be always T, for G C, and for C there will be G complementation, and for A there will be T A, and for G C again C, there is G, and for A T. This must be the complementation. 5 dash is there means there will be 5 dash here and then 3 dash here. If I'm going with this sequence, option A will be right. Only here A is extra. So the right option is option A. Next question, 96. Label A, B, C, D, E of given diagram. What is A, what is B, what is C, what is E, and then D. Here A is promoter, B is structural gene, and then C is template strand, and E is terminator, and then D is coding strand. So the right option is option A will be. 97. Promoter is located first at 3 dash end. Promoter is located at 3 dash end. Second one at 2 dash end. And then third one upstream of the structural gene. Fourth one downstream of the structural gene. So here promoter is located at 5 dash end, and then they are upstream structure, not the downstream. Downstream is for so here option B is right. Next, for the terminator, that is 98th question. Terminator is located at, yes, at the 3 dash end, not at the 5 dash. Upstream of the structural gene, no, it's a downstream. So here the right option is C. So the questions from transcription unit and then gene. 99th question. A gene is defined as A. Functional unit of inheritance, non-functional region of the DNA that hasn't any information. C. A and B both. D. None of this. Here the right option is option A. 100th one, cistron. And then last question of today's session. We will be continuing in the next part, part B, because this is a big chapter, as I said, and very interesting chapter also. So I made it 2 parts. Okay. Cistron is A. A segment of DNA coding for a polypeptide. B. Segment of RNA coding for a polypeptide. C. Segment of DNA that are non-coding sequence. And then D. Sequence of RNA that not any coding sequence. Here the right option is option A. It's a segment of DNA coding for a polypeptide. So thank you for the today's session. We will come back with the next part in the next session. Okay, thank you one and all.